can introduce.
I'm also recording it.
Wow.
Yeah.
Okay. So it's a real pleasure to introduce Sam Melton. So as we measure more and more data, particularly high dimensional data, let's say lots of details of every individual, analyzing such data is exceptionally challenging. And there are novel challenges that are, one could call the problem to be solved in the current times. And Sam has really taken them on, and he's going to tell us about both the challenges and the solutions he's come up with. It's been a real pleasure working with Sam. I'll really miss our scientific arguments and working together. Sam is also extremely stubborn in just the right way. So I think he has all the makings of a good scientist. This is the second defense from our lab in as many weeks. And defenses are bittersweet even in good times, but in these times, they're particularly challenging because we can't be together and celebrate. So I'm hoping that when the clouds lift, we get together and really celebrate Sam's success, assuming that he passes today. So Sam.
Thank you, Sharad. I would say I'm not as stubborn as Sharad might lead you to believe, but I did want to start off my, applied math is an inherently interdisciplinary subject. And so I wanted to start off my defense with a brief multidisciplinary explanation of what I've been up to for the past few years. And I'm going to do that through the form of a graphic, a pictorial representation of my PhD. And as you can see here, Sharad is sort of guiding me as I learn how to cut up a mouse with a commercial jet. And the meaning of this, more on this later, this should become clear. But now digging into this talk will generally be about data analysis and inference, as Sharad indicated. But the setting in which we're going to sort of view that is in developmental biology. And in developmental biology, the outstanding question is how a single cell, as shown on the left as a fertilized embryo, can give rise to some complex organism like a mouse or a human. And this process doesn't happen overnight. This process happens over a series of steps where the cell divides and creates a population of cells which all choose to downregulate or upregulate different genes. So they're changing their internal machinery in order to create all the different cells and tissues in an organism. And this happens over a period of days. Here you see a mouse embryo developing over about 14 days. And it's become fashionable recently to take these samples at different time points and grind them up so that we lose information about where each cell came from physically, and then sequence the RNA in each cell. So this gives us a quantitative description of the internal state of each cell. And so for one cell, I would have D different genes, and I would have some quantitative measure of each of those genes. So I would have D numbers that describe the cell. And I can do this for many different cells during different time points and from different tissues throughout development. And I get many different descriptions of all of these cells. And this is the sort of data we're interested in understanding. So we have M cells or M data points, and for each of those, we have D measurements about that cell. And this is what Sharad alluded to in the introduction, that this recent technology has given us these higher and higher dimensional descriptions of the cells. And there's some very basic developmental biology questions that we want to be able to use this data to answer. So we can sort of imagine graphing these cells as points in some high dimensional space. And so we have a sort of toy scatter plot here where my points correspond to individual cells, and you can see they lie in different places in some vector space defined by the genes. And we want to ask, first of all, what are the different states that are present in the data we've collected? So here I have cartoonishly drawn 3 states, and I would like to be able to color them. So I have green and purple and red, and these should ideally correspond to physiological differences between the cells in the system that we gathered the data from. But a second key point of this is I want to understand what defines these states. I want to know which genes are driving the differences between these states, and I want to know how these expression differences lead to physiological differences in the cells, so I know what role they're playing in the developing organism. And then lastly, I also want to understand the transitions between these states. As cells undergo development, they start as one population of cells, and slowly that creates all of the different cell types in the body: neurons, skin cells, lung cells. And I want to understand which of these transitions are allowed and what are the dynamics of the system as it progresses from a single cell to all of the cell types in a body. So we've learned a lot from developmental biology about how we should maybe approach this problem. And so in particular, I can go to a single time point in this trajectory that I've shown you. And people have found that by staining or looking at the expression of just a few key transcription factors, we see distinct populations of cells. So here I've colored some purple cells and some green cells. And the important thing to note here is that the cells seem to lie in one of these two camps. We have purple and we have green. And people found these genes through some sort of extensive experimentation and trial and error. But from here we can, let's see, we can see that at this one specific time point, we seem to have two different populations of cells. And then as we move on to the later and later time points throughout development, we see a variety of other cell types arising. These are the muscle cells and the heart cells that I referenced earlier. And further from developmental biology, we know that these distinct populations have distinct lineage potential. So for example, the purple cells on the graph here can only become these orange cells at the end of the day. The purple cells generally do not revert back and become the blue cells or the my skin color tone cells. And the lesson here is that there are these distinct populations, and the existence of these populations also corresponds to the developmental potential of the cells. So now the question is, going back to these data I referenced earlier, is can we take this data and understand this entire process, understand the presence of the different states, and understand the transitions that are possible between them. As a brief motivation for the perspective we're going to take on this problem, I wanted to reference a very powerful idea from physics, which is using order parameters to understand very complex systems. So if you have a system with many different degrees of freedom that are all coupled together, like a spin system shown on the left, there's been tremendous success in using a very low dimensional order parameter here. In this case, it would be magnetization, to understand the different phases of this system. And this has been useful both for measuring as well as for modeling the underlying system. And so the question that I want you to keep in your head throughout the rest of the talk is, can we find order parameters that describe a similarly complex system of genes regulating each other and cells transitioning between cell states? So ideally, we could find some low dimensional order parameters that describe the transitions between cell states during development. In part one, I'm just going to focus on two of the three questions I posed at the beginning, and then later we'll get down to number three. So these first two questions, we're just going to talk about what the states are, and we're going to talk about what defines the states. So experimentalists have, as I said, been gathering data from many systems like this, but we're going to focus on one for the next few minutes, which is a few days during mouse embryonic development, where a bunch of different tissues and organs start to arise. And the data from this is sort of, you sign up to be a PhD student, and they send you this NumPy file, and it has some data matrix like this. And so roughly in these situations, you're gathering hundreds to thousands of genes from each of tens to 100,000 of cells. And so in this specific case, I'm going to focus just on transcription factors, because those are the genes that drive the expression of other genes. And so any sort of indication in terms of the internal identity of the cell should be reflected in those transcription factor expression levels. And from here, we want to take this data again, and we just want to understand the different cell states that are present in the system. So the first thing people often do in this case is you have your high dimensional data, and you want to be able to visualize it so you can understand what type of cell states might be there. So a popular technique for doing this is UMAP. And what UMAP does is it constructs a graph in the d-dimensional space of all genes that just you get by comparing distances between individual data points. And then it tries to embed that graph into 2 dimensions. And so this graph ideally should reflect something about the distances in the high dimensions, but then give us a 2-dimensional representation of it. As you can see, though, there's sort of a problem. We can't necessarily look at this mess and understand the existence of the different cell states. And if you don't like UMAP, that's probably smart, but you could also look at some other sort of representation of the data in the d-dimensional space. And so here I can just look at cell-cell correlation, or the similarity between cells in the full dimensional space. But again, it's difficult to see the presence of all the cell types we know exist. So here we have multiple days during development where we know there are dozens of different cell states that have different developmental potential, and yet we can't see a signature of this by looking at the data. In particular, we can't see these two key states that we know of that are defined by two transcription factors which have very clear expression signature in the very early states. So why can't we find these states? What's the hang up? What's the disconnect between these high dimensional representations and the experimental reality? One potential reason for this is that the data is just too noisy. Maybe there's technical noise in the experimental setup that's preventing us from resolving the signal. And in that case, you're only as good as the data. So you're sort of up a creek with very few paddles. The second reason that we might be losing this is because of low data density in high dimensions. So data density or sort of how well populated our space is statistically is going to decay exponentially with dimension. So in high dimensions, we're going to be underpowered to run statistical tests. One third reason could be that maybe I'm just doing dimensionality reduction wrong. Maybe UMAP isn't the magic they would want you to believe, and the signal of the individual cell states is getting lost in this dimensionality reduction. But the last reason, which is related to all of these and is what I'm going to focus on for the next few minutes, is that the signal may be localized in a subspace with dimension much less than the total dimension D. So in other words, maybe only a few genes are important, and yet we're using all of the genes to measure distances. So what does this mean in a very simple situation? So here in a two-dimensional situation, I can show you one dimension, which is the x-axis here, has a lot of signal in terms of the cell type. So here I have green cells and I have blue cells, and I've just colored these in, but I've just generated this data. And the x-axis has a lot of information about which cell type or which cluster each data point belongs to. So if the x-axis value is greater than 0, it's likely to be blue, and if it's less than 0, it's likely to be green. The y-axis, on the other hand, which I've labeled a noise dimension, has no information. The distributions of each cluster are indistinguishable. So here, I would say only the x-axis is informative or relevant. And as I move to higher dimensions, I can sort of extend the logic of this problem. So in 3 dimensions, I can similarly I have my blue pancake and my green pancake that are coming out of the screen, sort of, and they're separated by 1 dimension. They're separated by this signal dimension, which I'll call ds. So in this case, ds equals 1 because we only have 1 signal dimension. And then here I have 2 noisy dimensions. So the pancake sort of spreads into 2 different dimensions, and so I would say dn equals 2. Now, here's, again, just a 3-dimensional example, and you're going to have to bear with me as we go to higher dimensions that I can't plot directly on the PowerPoint. But what we're interested in is, as we go to higher and higher dimensions, if more and more of the dimensions are noise, or dn gets larger and larger, how does it become difficult for us to resolve the signal? And so here, if this ratio of noisy dimensions to signal dimensions is very small, so I have roughly as many noisy dimensions as I have signal dimensions, then I can do one of these dimensionality reduction techniques, and I can resolve the existence of different clusters or different cell states. However, as I have more and more noisy dimensions, I lose the ability to resolve these differences. And it decays very quickly until it's all one of these spilled coffee graphics that I showed you that sort of resemble the mouse data that I showed you earlier. And one question is, can we look at data in this regime of very large d over ds? So we're in a situation where we have a lot of noisy dimensions, and can we find a way to move back? Can we find a way to eliminate the noisy dimensions so that we can slowly start to resolve the signal using more conventional statistical approaches? And this problem would be somewhat easier if we knew the clusters. So if I knew which data points belong to which cell type, then I could solve this. And I'll explain that. So here I have my green pancake and my blue pancake already. And I mentioned that the single dimension has high mutual information for these 2 clusters. So knowing this, I could just look at the dimensions that have high mutual information with my cluster assignments, and then I could use that to find my informative dimension. Conversely, if I didn't know the assignments of the clusters, but I did know the important dimensions, then I could take this high dimensional mess, here's a UMAP of some points where I don't know which cluster they belong to. I could project into the subspace that's defined by my important dimensions, and then I could start to see signature of my different cell types. And then I could color them in using a clustering algorithm that I choose. So the problem here is that we don't know either. We don't know the informative dimensions and we don't know the clusters. So it's unclear how to understand both of them simultaneously. And so in this case of the mouse data, the points aren't colored. They're all blue. I don't yet have purple and green and red points. And so I need to figure out a way to identify the informative dimensions or the clusters so that I can solve both of these. So the goal here, I'm going to focus on the dimensions. So I'm going to try to find a set of weights corresponding to my different dimensions that are informative for clustering or tell me some information about the identity of the data points. And so in this case, where I have my two pancakes, they don't have any different color here. They're both blue because I don't know the identities of the cells, or I don't know the identities of the data points. And I would want the E1 axis to have a large weight because that has information about clustering. And I would want the other two weights to be small. But I want to do this without knowing the number of clusters or their assignments. So I want to do this sort of in a way that's agnostic to what the clustering is of the data points. One question you could ask is, why am I focusing on just individual dimensions? The separation between clusters could be some distributed linear combination of many dimensions, or even some nonlinear function of many dimensions. And it's a fair question. Look, one reason why we think it's just a small set of dimensions is that clues from all of the experiments done in developmental biology have taught us that a few genes are sufficient to resolve the existence of different cell states. One reason from statistics is motivated by the success of compressive sensing in recovering sparse or recovering underdetermined signals by imposing some sort of sparsity constraint. So we'd like to impose a similar sparsity constraint on our search for important dimensions. And the last one is just experimental follow-up. So when you do any sort of computation, it's never worth anything unless Dennis can go and tag the genes and do some experiment otherwise. And if I tell Dennis he needs to tag 100 genes in a specific ratio in order to resolve the populations of cells, he's never going to do it. So the plan here is we're going to try to first construct a toy example out of synthetic data where I'm going to generate data where I know the answer. And I'm going to try to make that as hard as possible so that when I solve it using some sort of algorithm or approach, then I can go back to the single cell data and I have more confidence in my solution. So what would be the hardest setting? The first condition I want is I want a large D over DS ratio. I want a lot of noisy dimensions. This is just overall the problem I want to solve, but I want to make it a little harder than that. And so in addition to that constraint, in addition to having D over DS be very large, I also want my data geometry to be a little confusing. And I'll explain very clearly what I mean here. So here's a three-dimensional example of informative dimensions. So each of these dimensions is a relevant or signal dimension. However, each of these dimensions is only relevant for a pair of clusters. And so if you look at the first dimension here, E1, E1 is only relevant for distinguishing the red from the blue clusters. The green points, on the other hand, have some broad uninformative distribution in E1. Similarly, E2 is only informative for separating the green and the blue points. It does not separate the red points. The red points are uninformative in E2. And for E3, the green and the red points are separated. So the point here is that each dimension is informative, but it is minimally informative. It only gives us information for a pair of clusters, not all of the clusters. And so this means that if I looked at the marginal distribution of one of my signal dimensions, I would see signature of some sort of separability or some sort of signal if I knew the identity of the cluster. So here I've made histograms and I know which points belong to which cluster. So I can see the orange and the blue separate clearly in this signal dimension. So then again, if I knew the clusters, I could identify it. However, if I don't know the clusters, looking at this marginal distribution gives me no information because it's polluted by all the other clusters for which this dimension is uninformative. And this is the situation we're in with the single cell data. We don't know the important genes. We also don't know which cells belong to which cell type. So to review, we have this setting where we have a lot of noisy dimensions. And so we want to consider the problem in this difficult limit where the number of noisy dimensions is very large in relation to the number of signal dimensions. And furthermore, the signal dimensions are as uninformative as possible. They only have information for a pair of clusters. And the goal is to weight our different dimensions. We want to find weights to be able to later go on and project into this informative subspace of signal dimensions so that we can find the cluster. And I've said this before, but I want to step through this more clearly about how if we knew the state, so if we were able to color the data points into green, into blue, then I could construct weights that separate them. And I'll describe how I would do that very concretely right now. So if I had these blue and these green points, what I would do is I would build a classifier, which I'm going to parameterize by theta, which is normal to this plane. And this classifier is going to separate my green and my blue points. But further, I have this sparsity assumption. I have this idea that only a few genes or only a few dimensions matter. And so I'm going to regularize this classifier so that it lives in a low dimensional subspace. And here you can see that I've drawn this simple example very cleanly so that you can see that the vector normal to this plane lives entirely in the signal dimension. And so now I could go through and I could look at all my pairs of clusters and I could sum over my pairs and weight each dimension according to how much each classifier lives in that dimension, given the regularization. But again, this is a function of the cluster assignment. So this is a function of knowing that I have blue clusters and I have green clusters, which I don't actually know. So if I don't know the states, if I don't know which color the points belong to, I have some joint probability distribution over my weights and my cluster assignments. Because again, if I knew one, I could maybe guess the other, but I don't know either. And the solution we came up with is to average over the clustering assignments. And that's because we don't know how many clusters there are, and we don't know the sort of distributions that give rise to these clusters. So instead, what we're going to do is we're going to make a lot of guesses about what those cluster assignments are, and then we're going to weight our dimensions according to how well they do on average over all those guesses. So we can't actually integrate over cluster configurations because that would be assigning each point to many different clusters. So instead what we do is we sort of sample from this to approximate this integral. So first we sample the clustering configuration. We make a guess about how to cluster our data points. And then given that guess, we take our best estimate about which dimensions or which genes are important for distinguishing those clusters. And so in practice, what this means is we cluster the data thousands of times. We create this ensemble of guesses about the clusters, and then we weight our genes according to how well they do in each proposal or in each instance of this ensemble. So step one, I'll take my data, I'll throw it into some clustering algorithm. And so here I could color my points based on my guess. And so you'll see I previously had 2 blue pancakes, but now I have a rainbow pancake. 2 rainbow pancakes. And this guess isn't completely right. It's not, I haven't fully discovered the existence of these pancakes, but I have some coarse estimate. Then I'm going to look at all my pairs of these proposed clusters. So here I'm just going to take 2. I have A and B, they're green and blue, and I'm going to build a classifier that separates them. And now I'm going to update my dimension weights. So here, this classifier separates them in the E1 axis. And so I would add some weight to G1, which is my weight for that first dimension. And I'm going to do this over, as I said, an ensemble of cluster proposals. So this is one instance of coloring in the points, but I'm going to color in the points many, many different ways and average over all of those instances. So now we want to understand a bit about when this is going to give us a good answer and when this is going to give us a bad answer. And to start on that journey, let's consider what happens if we take 2 clusters that belong to different pancakes. So here I have, as I said, this green and blue cluster belong to different pancakes. So I build my classifier, and this is a good one. This lives in the signal dimension, as I said. So I'm going to add some weight to one of the dimensions that I want to be weighted highly. This is a success. And I'm summing over all of my different cluster proposals. And so as I move along, I get another pair that belong to different pancakes. So again, I get a plane that lives in the good dimension, and I get one in the signal dimension. The error arises when I have 2 clusters, or I'm considering a pair that belong to the same pancake. And in this case, I'm gonna draw some plane that attempts to separate them. It doesn't do a great job, and it's going to contribute to my noisy dimension. This is going to add weight to my noisy dimension, which is not what I want. However, as I do this over all of the pairs and all of the ensembles of cluster proposals, you can see that I'm going to weight different noisy dimensions as I do this average. And so this plane is sort of rotating in my space of noisy dimensions. And this turns out to help us because this means that an average over the ensemble, this error is going to get distributed over all of my noisy dimensions. And so I'm going to have this factor of 1 over the number of noisy dimensions that is part of the weight that I'm adding to each of my noisy ones. So now let's see if we can go back to the simple case, or the difficult, not simple, the difficult case of the Gaussian data that I've generated before. So this is, again, the most complicated example. I'm going to go into a large d over ds ratio where the data geometry is extremely complicated and see if I can rediscover the structure. So here I'm pretending I don't know the identity of the data points. I don't know which cluster they belong to. And I also don't know which dimension is good and which one is irrelevant. And then by averaging over an ensemble of proposals, I can weight my dimensions. And so here I've just grouped them because I have generated the data. I know what the answer is. And you can see that we're adding more weight to the informative dimensions than the uninformative dimensions. So far, this is a good sign. And also, I can take these weights. I've sort of built myself a metric, G, and I can take these weights and feed it into some dimensionality reduction method. And from there, I can see I can actually very clearly resolve the existence of these distinct clusters. But we want to understand a bit more about where it's going to work and where it's not going to work. So first, I want to introduce 2 quantities. So we want to have weights for each dimension. And I could imagine the average frequency of these weights for my signal dimensions and the average frequency of these weights for my noisy dimensions. And these are just the rates of me making a good guess and the rates of me making a bad guess. And what we're interested in really is the ratio of these. It's kind of like our signal to noise ratio, how frequently we're going to guess a good dimension. And because we can generate as much data as we want, we can sort of generate this for a variety of different number of data points and different points in this phase space of different ratios of d to dn or d to ds. So here you can see this, again, is just generated from Gaussian data. So I know what the answer is. So I can compute this ratio of gs over gn. And as I move to larger and larger d over ds, my signal starts to degrade, but it's over 1 in all of these cases. So at least I'm more likely to pick a signal dimension than a noisy dimension. But this is a bit messy. And it turns out we can make better guesses about how this is going to scale as we change the parameters of our problem. So to understand this, the first sort of term in the scaling is related to these counting arguments I gave you before and the rotation of the planes in the dn noisy dimensions. So as I said before, the rate of our errors has a 1 over dn term, which is related to the fact that there's some sort of spherical, or there's a rotational symmetry in the dn noisy dimensions. So that gives us this prefactor, which is good news for us because it means that as we go to the large d over ds regime, the numerator is going to grow. And that means we're going to get more and more signal. However, this can't be the full story because this would imply that as you had many, many noisy dimensions, you could always add more noisy dimensions and get more signal, which doesn't seem to be true. And there's a correction to this. So to understand the correction, first you have to say the cluster proposals that we build are clustering in the full d-dimensional space. And so that means that when they conduct the clustering, they have access to some characteristic length scale in the full dimensional space, which is going to scale like m to the negative 1 over d. This is the average distance between points in this full d-dimensional space. But that characteristic length scale is going to translate into a volume in the relevant subspace, which scales like m to the negative ds over d. And so the clustering algorithm has access to this length scale, but what we're really interested in is how accurate it is in the relevant subspace, which is sort of encompassed by this characteristic volume. So my correction term to this prefactor is going to be some function of this argument d over ds log n. And f, I don't know what f is right now, but I know that it's going to be monotonically decreasing. And it says something about how accurate my cluster proposals are, or how likely they are to lie in the same pancake that I showed you before. And so I can also go back to my, my numerical plots here, and I can rescale my axes according to some of the scaling arguments I made. So first, I would like to rescale my x-axis according to the argument of f, d over ds log m, and similarly rescale my y-axis according to these counting factors. And you see that the points start to collapse onto the same line. And in particular, as you move to the right on this plot, which is the sort of regime where we would expect these approximations to hold, everything seems to fall into the same line. But the overall lesson here, and this is maybe one of the important takeaways of this section, is that every clustering or every analysis that you do in the full space is going to be taxed by this factor. The fact that as d over ds log m increases, you're going to have decreasing accuracy in understanding the true relationships between data points. However, by averaging over my ensemble of guesses, I get this prefactor, which actually grows as dn grows. And that's going to save me. That's going to allow me to make some good approximations, even in a regime where everything else isn't working. So now let's go back to the mouse data. Let's see if we can look at these high-dimensional single-cell expression snapshots and connect them back to experiments. So I can take these data, and this is again, this is 720 dimensions. So these are the expression of 720 different transcription factors in about 12,000 cells. And I can create an ensemble of cluster proposals or an ensemble of guesses, look at genes that separate pairs of those guesses, and then reduce that to a subspace of just 27 transcription factors. So this would put me in the sort of regime I would expect, where these figures of d over ds and d over ds log m mean that normal clustering algorithms shouldn't be able to pick up the signal in the informative subspace, but I should be able to get a pretty good signal to noise in order to resolve this. And if I look at my cell-cell correlation matrix now, you can see that you start to see some structure. So here on the left is the cell-cell correlation in the full 720 dimensional space. And then in the center is in a 27 dimensional space, which are just the transcription factors I found to be important. And you start to see signature of different cell states. So this is very encouraging. Similarly, you could look at UMAP if you insist, and you can see that there are also, you start to see the existence of these different cell types that are sort of splitting off, or at least different clusters in the data which could be cell types. But these dimensionality reductions can still be misleading. We're still looking at 27 dimensions in a 2-dimensional projection. So what's perhaps better is to look at the identities of the genes and use our good friend Dennis, who has extensive knowledge of gastrulation and early mouse embryonic development, to understand what these genes actually mean. And when we actually look at the names of the genes, we find that these are sort of a laundry list of important genes for early embryonic differentiation. So we can see the clear existence of different cell types that biologists have already identified and known existed, and they've been staining for these factors for years. So that indicates we're on the right track. That indicates we're finding the sort of genes that we want to be able to find. In particular, you can see these two genes I had brought up earlier, the purple and green. You don't need to remember the names, but these are two of the highly weighted genes. And furthermore, we were able to find subpopulations of the epiblast, which were not actually identified by the original papers that published this data. And these populations are important for the sort of the actual development of the tissue, because we found populations that secrete specific factors, which then guide fate decisions in other cell types. So to summarize so far, the goal was to take this high dimensional data and essentially divide the data points into clusters and find out which of the features of the high dimensional data define those distinct clusters. And first, what we learned is that some of the structure of some of the signature of these clusters can get lost in high dimensions. But by looking for some sort of sparse signal, not only can we make serious steps statistically, but also experimentally. Because now I have a list of genes that I can go and do an experiment and tag or stain for and see what they actually do in the embryo. So now I'm going to move on to part 2. I want to quickly check if I can't see the chat, but if there are any questions, I could pause for a second for questions. Is there a chat? There is a chat. There are no questions. Okay, I'm going to move on. And part 2, I'm going to talk about, oh, there is a question. Let's see. Leon has a question. Leon's name is also on this slide.
Can you hear me?
Yes.
So there's a bit of a debate in the developmental biology community about whether cell states really exist or not. And you know, there's one line there's one line of thought that essentially says that cell states are sort of a social construct, just like races and binary gender.
Yeah.
And that it's all sort of a fluid continuum. And so in your work, you have been assuming that these cell states do exist. But what if they don't exist? And what if the signal dimension, that there are no relevant signal dimensions? Are you just sort of, is your model sort of just imposing this on the data or not?
So I think Leon's baiting me a bit, but I'll take his question earnestly. So it is quite common these days for people to construct some sort of representation where they say they find the cells live in a continuous landscape or something like that. So there are a number of things about this. One is that these techniques they use, they are trying to find a dimensionality reduction that reflects a continuous manifold in the high dimensions. So in that case, they're forcing their data to live on a continuous manifold and then projecting that in 2 dimensions and going, oh, look, it's continuous. But obviously that's what you're searching for if you're looking for a continuous representation in the high dimensions. The second thing I would say is, if I find separability between cell states in terms of transcription factors where I see 2 clean distributions, then regardless of what you do with your high dimensional mumbo jumbo, you can look at these one-dimensional distributions, and you can see not only is there a statistical signature of the difference between these cell types, but also if it's a transcription factor with very drastic differences in expression, then that would indicate some sort of biological difference as well, because transcription factors drive the expression of various other cellular processes. But at the end of the day, I think it comes down to, you do your analysis, and then someone is going to have to go and test this and see by staining or by tagging different cell types what you actually see, and whether these differences correspond to different developmental potential. And so I'll get to that in a few minutes. Thank you, Leon. Any other questions? I can't see the chat. Yes, no more questions. Okay. So moving on. So this work, I just thank Leon for the question, but also thank you, Leon, for helping me out in this next section, which we did a few years ago. So the goal here was to say, we already have these cell states. We already have a subspace of dimensions which we think are informative, subspace of genes which are important for the identities of different cell states. And I want to be able to take these cell states and reconstruct the sort of lineage dynamics or lineage trajectory that the cell states undergo as they proceed through development. And our strategy here was to take lineage trees where we know the answer. So take examples from the literature where people have verified the lineage trees and look at expression patterns of these key genes, and to see if then, so we have a sort of a lineage tree and we can look at the expression patterns of these individual genes. And the hope is to look for some sort of pattern or signature that we could then use to go back and look at the distribution functions of each gene to infer the lineage relationship between the cell types. So when we're looking at a perhaps complex lineage tree, we're going to first start by dividing it up into triplets, or just groups of 3 cell types each. And this is because with 3 cell types, I have the minimal subset in terms of number of cell types where I have a non-trivial topology. So with 3 cell types, I have A, B, and C, and I can only have a few different topologies. I could have A as the root and B and C are the 2 leaves. Or I could have B as the root and A and C are the leaves, or C is the root and A and B are the leaves. And these are the only topologies that could really happen in development. So one could say, oh, you could also have a loop, but generally in development, we don't have many loops. So we're just going to say that you have transitions, sort of sequential transitions in one of these topologies. And this also means that the topology of a triplet is completely determined by identifying the root. Because if I know the root, I can draw in the triplet, and I know the topology of this sort of subtree. So if I want to identify the root, and I want to look at individual patterns of gene expression amongst these 3 cell types, there are only a few different distributions that I could actually see. So if I have the distributions of these 3 cell types, and I'm looking at the expression of a single gene, one example of a distribution function I could see is one cell type being minimum and two cell types having higher expression, which is shown here. So here, one cell type has lower expression, the other two have higher expression. Another example of a pattern I could see is a clear maximum pattern. So I could also see one cell type have a maximum and the other two have a minimum pattern. And these are the only two patterns I could see in these informative genes that have a signature of separability between the cell types. So you could also say, oh, you could have three separate distributions, but that would fall into both categories because then I would have both a clear minimum and a clear maximum. Now, given one of these patterns, the important thing, as I said, is we want to identify the root. So we want to be able to see where in this space of distributions the root lives. And if I have the clear minimum pattern, I have two possibilities. Either the minimum, this clear minimum, is the root distribution. So the cell type that is the root has expression of this gene, which is much less than the other two cell types. Or that clear minimum could be one of the leaves. These are the only two possibilities for identifying the root with the clear minimum pattern. Similarly, if I have the clear maximum pattern, I could have that maximum be the root. So the expression of the root is higher than the expression of this gene in the root cell type is higher than the other two. Or I could have one of the leaves be the clear maximum. So what we did with Leon is we looked at known lineage topologies, and we tried to see which of these patterns correlated with the known trajectory or the known topology of development. And what we found is that the clear minimum was what actually correlated with the developmental trajectory. And so what I mean by this is that when we looked through the expression of individual genes, we very rarely saw a clear minimum pattern where the minimal expression was in the root. Rather, if we saw the clear minimum pattern, it was much more likely that the clear minimum was in one of the leaves. And so this gives us sort of machinery for starting to understand these lineage triplets, because we can look at the triplets and we can classify genes according to the clear minimum pattern and use that to infer the topology. So to describe clearly what this means, if I have one of these triplets that I looked at where I know the topology A, B, and C, where A is the root, if I saw genes with the clear minimum pattern, I would see many genes like this where the minimum is in B, and I would see many genes like this where the minimum is in C, but I would not see many genes where the minimum is in A. And so that also allows me to sort of club my genes together. I can take a group of genes which have minimum in B and a group of genes which have minimum in C and start to visualize my triplets, or visualize the trajectory of my triplets, where I would have A going to B or A going to C. And one thing to note is that this sort of pattern of having up, up, down in two classes of genes is symmetric, is consistent with the motif of mutual inhibition, where I have these two classes of genes inhibiting each other, which would lead to three stable points, or a bistable switch. But more sort of concretely, what we want to do is use this pattern to infer the lineage of a triplet where we don't know which one is the root. And we can do this by using Bayes' theorem, by looking through our genes one by one and classifying them to see if they fall into the clear minimum pattern. So here I have X, Y, and Z, and I don't know which one is the root. I could look at the first gene, and I see this gene doesn't have a clear minimum. So it has, these two distributions are both very low, and this one's very high, so that's more of a clear maximum pattern. I would not consider that for the inference. Similarly, for the second gene, these three distributions all seem to fall on top of each other, so that doesn't give me any information about it's not a clear minimum pattern. But now I go down my list of genes and I come to this gene, gene J, and gene J has a clear minimum in cell type Z. So it is clearly downregulated in cell type Z versus X and Y. And so I would remember that gene. That gene is going to be important for inferring the lineage relationship. Similarly, I can go to gene K, and I see gene K is also, I see downregulation in cell type Y and higher expression of this gene in cell type X and Z. So together, these two genes give me guesses, because I know that the minimum in these genes is unlikely to be the root of the topology. So from here, I can infer a probability distribution over my topologies. And in this case, this sort of toy example, I have these two genes which are voting against Z being the root and voting against Y being the root. So it's more likely that cell type X would be the root of this topology. And we use this, this sort of pattern and this inference algorithm to understand a bunch of lineage trajectories that we couldn't understand previously. So this is an example that we did with our collaborators at the Allen Institute, where they sequenced cells from an in vitro neuronal developmental system. And we were able to find signatures of the different cell types that arise in the brain. And furthermore, we were able to predict these splits. So you can see the split right at the top, where one branch leads down to the neocortex and the other branch leads down to the midbrain. And these splits, not only did we sort of get this tree to understand how the different cell types arise, but also they were able to experimentally validate the different branches of this tree using viral barcoding. The second thing we get from this analysis is we find these key genes at each transition. And these are the sort of order parameters that I referenced earlier. Because if we look at one of these transitions from C0 to C1 and C2, this split into the different cell types in the brain, we can plot them, as I said, in these axes that we've discovered. So we've discovered that these genes DMRTA1 and CEBG are both indicative, or they both allow us to measure the identity of the cell as it's progressing through this sort of decision-making process. And these order parameters are actually very helpful experimentally. Not only can we make those plots to understand the topology of the tree, but also colleagues in the lab have used these to measure cells in real time as they undergo cell fate decisions. So here, Lance, who's a postdoc in the lab, hopefully is watching, he took a collection of stem cells, which are like the epiblast cells, or the very early pluripotent progenitors in mammalian cells. Here he has human stem cells in a dish, and they're fluorescently tagged. So you have green corresponding to one transcription factor, SOX2, and red corresponding to another transcription factor, OCT4. And what we would expect is as these cells transition down one of these branches, so as they make the decision about whether to become neural or mesendodermal, they should turn off one of these transcription factors. And so here I have this movie, courtesy of Lance, and all the cells start expressing both green and red at the beginning. And now as I play the movie, you'll see cells start to downregulate one of these factors. And this is us watching in real time as these order parameters indicate the progression of the cells along these different states. So you can see the cells in the middle seem to become more neural, and the cells around the edge seem to become more mesendodermal. And that's it. That's all I've got. So to conclude, the important take-home message here is that when you're dealing with this high-dimensional data, you have to be very careful just gathering more and more dimensions or more features about your individual data points doesn't always help you. In fact, it can just pollute the signal more and more, so you lose the signal. However, by averaging over these different cluster proposals, or by averaging over sort of bad guesses about what we think the structure looks like, we're able to find these important dimensions. And lastly, these dimensions are not only statistically important because they allow us to find the existence of different cell states, but they also allow us to understand the transitions between the cell states and would provide a sort of obvious follow-up in terms of experimental perturbations and measurements. And I'll take questions in a minute, but before I take questions, first, I really want to thank Leon. When I started grad school, Leon was a senior and veteran grad student who helped me a lot, both navigating grad school and also sort of shaping the sort of questions I wanted to go after. I was very lucky to have him in the lab when I started my PhD. Also thanks to Dennis. Dennis, whenever I have a list of genes, I go to Dennis, and Dennis understands all the biology, so that's important to have, and so I don't make any silly mistakes. Vilas was a collaborator at the Allen Institute who helped with a lot of computation and the experimental follow-ups that I mentioned earlier. Suman, I don't know if she helped with anything directly, but she was a member of the lab who I was very appreciative of her sort of help early in my grad school experience as well. Also Lance, who gave the video I just showed and was a great office mate for the past few years. And all of the Ramanathan lab, I have an old picture. I think only maybe half of the people are still around, but everyone in the Ramanathan lab was helpful for giving feedback and discussion along the way. And also Chris Stody, who is just always, like, whenever I make a mistake, Chris helps me sort it out. Also, thank you, Cengiz and Ariel. Cengiz has been very friendly and helpful in terms of giving feedback on my work over the past year or so. And Ariel similarly. Ariel is sort of a role model of mine because he seamlessly transitioned from statistical physics to biology and has been going after some interesting biological questions. So it's good to have people like that to look up to. And lastly, Paula Arlada. I'm not sure if Paula was able to make it, but Paula has always been so nice and supportive and answers all my questions and is extremely helpful throughout grad school. I was intimidated by her at first, but she's been so nice, and I really appreciate all of her support through grad school. And also Chris Rycroft, who I've had the pleasure of talking to a lot about different areas with data analysis and stuff like that. And then, of course, I have to give a big thank you to Sharad. All of the ideas in this presentation are the product of many discussions with Sharad. And beyond him being a incredible scientist, he has supported me undeservedly throughout grad school in so many ways. And every time I mess up or an administrator gets mad at me or I send an email I shouldn't have, which happens far more frequently than I'd like to admit, Sharad is always there to kind of have my back, and he doesn't even ask me to apologize, really. He just has my back, and I don't know what I'm going to do without him. And of course, I also want to thank my family. My parents on the left have been the only people more supportive than Sharad in the whole grad school experience. And they've just made it so easy for me to focus on science because they help with everything else. And you also see my dogs and my sister and Brad, her husband, and Ben. This is the only good photo I found of Ben, and we don't have many family photos, but I'd just like to thank my whole family for all of their support. And now questions. If Roya says LOL, but if we have any other questions, this would be a good time before we disband.
Hey Sam, this is Sean. That was a great talk. In the first part, when you're talking about clustering,
yeah,
to what extent do you get clusters that are real, biologically real, but not the kind of developmental cell type clusters that you are really looking for? You know, clustering that's driven by other things, circadian rhythm, metabolic state, stuff like that. How much does the other kinds of biology confuse what you're trying to see?
Yeah, so that's a great question. In general, I mean, you can take the data and you can cluster it, but I don't know what that answer means until you sort of look at the specific genes and see what they mean and see whether I can map those to known biological differences. One thing we do to ease with that is that I'm only looking at transcription factors. So any signature of separation that I see in the space of transcription factors, I would argue is at least somewhat biologically meaningful because the transcription factors are likely driving the expression of other genes. But then again, at the end of the day, I think you still have to take the clusters and do some sort of staining or some sort of experimental follow-up. Otherwise, I don't know if there's any good way to validate the specific clusters. So you also mentioned circadian rhythm or something like that. This is, I think, a common thing. Some people sort of weed this out beforehand by taking genes that correlate with that and neglecting them in their downstream process. I've done it both ways, and I haven't noticed much of a difference. That's not something I pull out in my in looking at transcription factors. But that's a good question. It's not immediately obvious why I don't pull it out, I guess, because that might be sort of that seems like you should get some sort of bimodal signature of the circadian factors or cell cycle or something like that. But if that difference exists, it's dominated by other developmental differences in the gene expression data that I see more prominently. Any other questions?
Yeah, can I? Oh, so there's one in the chat if you want to take that first.
Chat. Why do we assume that the clusters are linearly separable? What does that correspond to biologically? So a good question. So as I said, that it could be that there's some nonlinear separation between the cell types. And if this is true, then we're sort of in trouble because we can't actually go and validate that experimentally because it's some sort of complicated relationship of genes. And what all of these clusters I find doesn't mean that there isn't some sort of nonlinear separable distinction between cell types. But what I'm going to start with is just finding the linear separable signatures. And then if there's anything nonlinear, that's sort of a separate task for us to confront later on.
Can I ask about this pattern that motivated the second part, the observation that the leaves tend to have the clear minimum expression? Like biologically, what's going on there? It feels kind of reminiscent of like you turn off a pluripotency factor to differentiate. Is that what you think is going on? It's kind of striking that it's always true, seems to be your assertion.
Yeah, so, well I mean, it's not always true. I mean, there's some sort of, there's certainly probably situations where you get it wrong, and there are situations where it wouldn't be as clear of a signature. But it seems to be more often than not. The picture we have in our heads, which again may or may not be true, is this idea of mutual inhibition. So if I have two classes of genes and they inhibit each other and activate themselves, then that would lead to a bistable switch where I have one fixed point, one stable point, where both of them are on, and then two other fixed points where one of each gene is on and the other is off in each of my two fixed points. So this is sort of a hand-wavy explanation for why we might see this pattern, because if we had mutual inhibition at each of these sort of bifurcations along a lineage tree, then you would expect a pattern like this in some genes. As I think you mentioned, this is also somewhat consistent with like, I think another kind of hand-wavy hypothesis people have talked about during development, which is the idea that in the pluripotent state, you have a lot of things that are on, and then you sort of turn things off as you go down. But it is also important to keep in mind that I'm just talking about a few genes here. So in the pluripotent state, I have maybe a set of a few genes which mediate the first transition. That doesn't mean that all the genes are being turned off, because other genes could be more gradually coming on, or they could just not fit the clear minimum pattern, but they could be being turned on down either of the branches, if that makes sense. So as you move along the developmental tree, the important axes you want to look at are going to change, which means that this doesn't mean that genes are always being turned off as you go further down in development.
Sure. Okay, thanks.
Yeah, thanks for the question. Any other questions? All right. I think that's it then. So yeah, I don't know. Everyone can get up and leave, but
We have to applaud first.
Oh, you have to applaud first. Thank you. Yeah, Charles has already applauded. Oh, this is great. You guys are fantastic.
Well done.
Thank you all for coming. It's great. There's so many people here. I'm just feeling now. Usually, not this many people come to my talks. So this is maybe this is the silver lining of everyone being at home. I get more people to show up. So yeah, thank you all for coming. I think now Ariel and Cengiz, we have to separate out somehow.
So we just have to leave the meeting and then we can move to the other link.
Okay. Ariel and Cengiz, do you both have the other link? Thumbs up. Great. Okay, I'll see you all in a minute. Thank you all for coming. Yeah.
Your little hat.